Hello and welcome everyone in another episode of Researcher Celebrity. Today we have Dr. Dhananjaya with us. Dr. Dhananjaya has done his BSc in Life Sciences from Utkal University, Odisha. And then for Masters, he chose Industrial Biotechnology at Berhampur University, Odisha again. For PhD, he moved CSIR IACT Hyderabad and moved to USA for his postdoctoral research at Moore's Cancer Center, UCSD. And presently, he is a junior faculty at Levine Cancer Institute, Wake Forest University. With this brief introduction, I would like to welcome Dr. Dhananjay. Dr. Dhananjay, welcome and thank you for being our researcher celebrity.
Thank you, Dr. Khuswa. Really, it's a pleasure to meet you again after a long time. We met at UCSD and we stayed together for I think a couple of months. So yeah, nice to see you here again. Yeah, before start our conversation, I would just like to add few points here. That yeah, I've seen now is the arena of digital area. So every people used to put showcase their talent, even if it is small talent, they put it in the YouTube and whatever. Maybe. So I was thinking it was in my mind, since long days that why someone is not attempting when it's from a scientific background person, why not someone is not attempting to do, to understand what the PhD student or what the researcher is going through in India, especially the PhD students or who are going to pursue their career in scientific area. So and I appreciate your effort that you made it, and I was so happy that after seeing your foundation that is doing good, and this a lot of people are coming to your platform and giving their views, opinion on their research, and I think definitely it will helpful for the next generation or like a future student who come to research.
Thank you very much, Dr. Dhananjay, for your kind words. And at Empowering Science Foundation, we believe in breaking the barriers. As you mentioned, this is one of the barrier that people don't see researchers as celebrities. In the real life, we are the heroes. The researchers are the heroes who do all of it, be it fundamental research, be it translational research. So our due, our contribution to society, what we are serving here is celebrating the research, and that's how we named it as researcher celebrity. Thank you very much for your positive words about the foundation and this series. So let's start the conversation that how and when you decided that you wanted to be a researcher.
It's a good question, and I've seen you are asking this question to all of these celebrities. So uh, my journey will be a little different, and definitely I will share with you, and I will definitely your viewer will also look at this. Yeah, so I came from a middle class family, from middle class family where my father was teacher, school teacher, my mother is a homemaker, and all these middle class family, they always given priority to the study, you know they want their kids or child to make their career well, by uh, being a doctor or maybe engineer or maybe something higher to IAS or anything. So mine was not the same case. Mine, I was though I was well in school days, like in studies. Uh, it is somehow echoing. I don't know. So yeah, so my parents never put pressure on me that I will do this. I have to be an engineer. I have to be a doctor, or I have to be something else. To be very famous in the society or something. They never put any pressure on me to do that. Uh, and second thing was like we didn't get much exposure when we are at village. So you will see your friends, they are involved in different things. So we are also on the same category. I used to go for play. I was good in cricket, but yeah, but I was like jack of all trade, master of none kind, you know. So life was going like this. So once I you know crossed my 10th, 12th, and I reached the graduation level. So in mid of my graduation, or you can say like end of my graduation, I try to understand the intrinsic the intrinsic part of the biology, especially the botany, the genetics, especially more in the genetics and botany sort of thing. And I will give credit to that my teacher, Mr. Pavitra Mohan Behera. He was my graduation teacher. He inspired me a lot to, understand the basic, just don't think much about what you will get from this, but understand the basic, what genetics is. Mm. What he will connect the genetics into the normal day-to-day life? How it the genetics involved in the agriculture field? As we are a botany students, we think about that way, and how it you know how you employ this genetics in like cultivation or some of the like botanical area. So that was his lesson to me.
Mhm.
So uh, so after passing my BSc, like the graduation, I used to think about the genetic engineering field. So I used to read the basic plant genetic engineering, and he always pushed me to just read this. Uh, that like the BSc, the once your graduation exam done, from like April to like mid of June, you have a empty space time. So in that time I used to like prepare myself to understand the basic genetics, basic plant genetics especially.
Mhm.
But fortunately or unfortunately, I landed into a different subject in my MSc, which is industrial biotechnology. The other thing I would like to say, that I had no uh, ambition to do research. I had no
Mhm.
ambition to be a great scientist or something. There was
Mhm.
nothing in that time. There was nothing in my mind at that time. Just have to you know get into MSc.
Mhm.
If we will find some job after MSc, we'll do the job, because the other thing is that I don't want to be a burden on my parents. You know,
Mhm.
so I want to earn whatever is there. I want to earn something on my own, and we'll
Mhm.
see what will happen next. I never planned anything in my career. I never
Mhm.
planned, but
Mhm.
think what I did, you know, little bit hard work, and always the door was open for me.
Mhm.
Like next, what you'll do next is you just go with flow kind of thing.
Mhm.
I never planned such a way to do. So after completion of my MSc, so I tried the CSIR exam, all these things. So that time I was not that confident, or maybe I was not well prepared, so I didn't crack that time the CSIR exam. So
Mhm.
I went to Hyderabad to get uh, you know uh, uh, take coaching on this CSR exam to qualify CSR net exam. So fortunately, the first attempt I able to qualify the first round. That time two session was there. One will be like multiple choice. The other time is like you have all these descriptive types of question. So
Mhm.
anyways, so first time I qualified the first, but I wasn't able to qualify the second. But still, I was trying to appear the exam repeat every 6 months. So in the
Mhm.
meantime, I got a notice in the sense, I got a notice in the newspaper somewhere that there is a project JRF position vacancy there in Indian Institute of Chemical Technology, CSR Hyderabad. So, and the position was regarding the biology, chemical biology related, chemistry and biology. So I didn't know anything. I just go through it. As I told you, uh, I need to be self-reliant. I need some kind of I need to stood my one leg. That's what. There is nothing what will going to happen in the JRF research. What research? Nothing in my mind. Very clean mind. Just will go and let's see what will happen.
Mhm.
So they called me for the interview, and the interview went good, and they select me.
Mhm.
Once they select me, they put me under one the boss, my PhD boss. Her name was Dr. Monica Palbhadra. Okay, so I didn't know anything about research, what they have done, what their background, what they are doing in their lab. Nothing, no. I was just happy that I'm going to get 16K per month. I'm very happy for that. I'm happy with that. No, no other thought of science, nothing.
Mm-hmm.
After joining there, I get to know that Dr. Monica Palbhadra and her husband, Dr. Utpal Bhadra, they are the 2005 Nobel nominated scientists.
Great.
They
Mm-hmm.
got the Nobel 2006 Nobel Prize got an RNA interference.
Mm-hmm.
Then I heard these things from the other colleagues. Yeah, they are big scientists. You came here, you have to prove yourself that you are the best. You come here. So then I thought that we'll see. Means everything we'll see. What will happen? It will happen. Let's see. Even the boss knows me that I didn't have much experience on her topic. So
Mm-hmm.
I'll get into it. I will learn and I'll do things what the way I want. But yeah, fortunately, she gave me a project, and I try to student is on her expectation. I learned a lot from her. Initially, she used to taught us everything, every single things. But the only one thing was in my mind that I always even told to my juniors also. I was a person, I never do things what others do.
Mm-hmm.
It whether it is like 99% wrong,
Mm-hmm.
1% right. But
Mm-hmm.
I will do what I will feel that is totally different what others are doing.
Mm-hmm.
So in this context, so my PhD fellows, they have joined with me.
Mm-hmm.
So they used to do because we are in a chemical biology lab, and IICT is known for chemistry, especially the chemistry. What we used to do that we used to screen the molecule, the chemistry guys they synthesize and submit to us to screen or evaluate those molecule in different cancer model.
Mm-hmm.
And in that scenario, eventually you will get number of papers on that, because you have to do screen, you have to do some the IC50 correction, IT50 correction, and do some cell-based assay. Immediately you will get some paper. And all my lab fellows, PhD fellows, they have like 10, 12 paper by screening those molecules. But I choose a different path. I don't want to screen this molecule. My boss forced me a lot. Why didn't you screen? How will you develop yourself if you do want to have a paper? But I didn't have interest on those. That's what I told you, that I had never from the beginning. Even I play cricket, I try to play a different shot what other people used to play. Whether I'll be out, whether I'll be out in duck, but I will play that already. So it is from my it is like in my mind always. So I choose a topic which is totally different. You know. Totally different from what my lab used to do.
Mm-hmm.
So in my BSc, as I told you, like graduation time, I used to read more about genetics. I tried to understand the basic genetics.
Mm-hmm.
So uh, it helped me a lot in my PhD.
Mm-hmm.
So like I try to understand the initially I try to understand the basic the chromatin structure, the role of this epigenetics in cancer development, especially in glioblastoma. And third thing to understand, because my boss background was the RNA interference, and try to understand the mechanism, how the RNA interference work, and how it help in developing cancer, and how this RNA, like siRNA or microRNA, mechanistically work in cancer progression. So that was totally different what others were doing. And I was really enjoying doing those types of work. And I
Mm-hmm.
used to do those intricate, the sophisticated biology experiment, like even other people used to do. That's I'm not telling that I only know those, but it was known for it was unknown for my lab that time. So
Mm-hmm.
I used to do all these protein-protein interactions, those GST pull-down assays, a lot of co-amino precipitation, understanding the intrinsic aspect of that biology, how it is working, always how it in mechanistic manner.
Mm-hmm.
Uh, you cannot believe that my I have like 6 to 8 PhD mates. their thesis is different, and my thesis was different.
Mm-hmm.
There's no match between these two theses.
Mm-hmm.
But yeah, eventually I also failed a lot of time, but I kept in my mind that I will do whatever others don't do, and I will
Mm-hmm.
definitely tell your viewers that this is the main aspect of science. I believe myself, do what others don't do.
Mm-hmm.
It be wrong, but the 1% you'll be right, that will be novel for you. I
Yes.
don't tell you will be novel, but eventually you will get something what which will be helpful, which will give you more satisfaction than copying something. Who is doing that? I will do that. Let's progress our career like this. I don't think that will add anything into the scientific fraternity or scientific career. I don't think so.
Mm-hmm.
And the other thing that I met a number of people in my PhD
Mm-hmm.
uh, whom I really admire. One guy is called Sudeep. His name is Sudeep Mukherjee. He was here in Texas, Rice University. He recently moved to IIT, um, mm, that BHU, IIT BHU, and he was so great. I think I have met one of the. He is one genius person, I can say. His thought, his scientific ability, his ability to understand the science. I think I'm fortunate to have him, my roommates or side roommate. Believe we used to have you know, mug of like beer in PhD and
Mm-hmm.
try to extrapolate crazy idea. You would believe. Yeah, I will
Mm-hmm.
just give an example to your viewers. So we discuss it too. Why the heart doesn't have a cancer? Every organ have a cancer, kidney, lungs, this, that. We just throw like crazy idea. Why this heart doesn't have a cancer? Does it have more ATP production? And does ATP more production has any role in this? So these are like we used to throw this, crazy idea. And we used to enjoy this. And he used to enjoy. He used to discuss with us. I used to go and discuss with us. So I think those kind of crazy ideas somehow help me in reach to where I am now.
Mm-hmm. Mm-hmm.
I'm not saying I am a big place now, but if you see a person who has no plan, scientific plan, nothing,
Mm-hmm.
but he came from a village to here, just go with flow and develop himself or herself as, you know, think with that idea, think that way, and reach to that stage. So I think, due to the uh, my graduation teacher blessings, maybe my PhD mentor blessing, the friends everywhere. So maybe I reached here today. So after finishing my PhD, I moved to the Moores Cancer Center
Mm-hmm.
and got a chance to work with Dr. Donald Durden. He is a renowned professor in pediatric oncology in US. Most people knows him, Dr. Donald Durden. The first day I can share with your viewers, the first day I met Dr. Donald Durden at UCSD, the first thing he said me that we are here to do something for the America. I was strange. Means a person is 70, 71. He's thinking about America and the well-being for the American.
Mm-hmm.
His small effort can give a little bit relaxable, you know, from cancer to the American people. So I may not share with you when I was at UCSD, but when I was coming home, I was thinking that what what is this, man? Means I was telling myself, what is this, man? You just came here thinking about that I will get 50K per year, having 4,000 USD, and I will immediately convert it into Indian rupees, and I will tell my friend, hey, how much you are getting? I'm getting this much. You are getting. And the other way, a man of 70 is thinking about, well-being for the American. He has to develop
Mm-hmm.
something which will, helpful, which will help for the common American people, those who are, having cancer. So
Mm-hmm.
that strike my mind. So that you have to, feed yourself something so that you have to think in that way.
Yes.
Yeah. So the science is not only will get some paper, you'll have a good future, and those are the other thing. But towards the humanity, a scientist must should remember that you should have what you are doing for people.
Yes.
mm, and what you are going to give, you are going to give a relaxation, from cancer to the people.
True.
So I was very interested when I talked to him. Like I I thought, yeah, maybe he's the perfect guy for me, who understand the basic need of the people.
Mhm.
So he just the first second he just gave me a paper, just go with this flow. It's not like that. PhD means it's a training for you. PhD means you have to deliver, you have to perform. Sorry, postdoc doesn't mean it's a training for you. Those people who said postdoc training, I think it's a bullshit. Postdoc, you have to deliver, you have to perform the day you reach the lab. And the PhD was your training. The
Yes.
good you are in training, you have to come and deliver. No one will teach you here. No one will spoon feed you
Mhm.
anything here at US. So he just gave me a paper to understand the scientific uh, background of his lab, and he just tell me that go with that. Is all the dex is yours. You just go with that. From that day, and you know that I used to go every day to lab. I come by 10
Mhm.
p.m. from lab, go at 8 and come by 10 p.m. So initially, I had to put effort uh, on his work. He got some grant. He's happy. Now still I'm continuing with him uh, till now. So it's like my journey. Maybe I left most of things in between, lot of things, but yeah, this is how I came from a village to here without any. There is no ambition till my MSc, nothing. So hopefully, this I think the people blessing, friends blessing, the teacher blessing. I'm here, nothing else.
That was very sweet and crisp, Dr. Dhananjay. And I would like to tell all of our viewers that most of our researchers are in present are coming from families which are not very well, equipped financially. So that's when Dr. Dhananjay said that he comes from middle class family. It is about the financial limitations. We can say that when a student who wants to perform or who want to pursue their own dreams, be it become a researcher, be it be anything, like they seriously want to help their families and don't want to be burden on them, as Dr. Dhananjay also said. So the motivation there is to earn as early as they can. But as you have heard till now, that Dhananjay has walked on a path where he wanted to, but it was just like at that time it was not realized that he wanted to be a researcher, because there are so many opportunities which you can do. And as Dhananjay also said, that he wants to do something which most of the people doesn't do, and research was one of them. Not most of the people want to come to research because finances is not a driving force in research. Research is about your passion, how passionate you are. And as Dhananjay's mentor has taught him this, now we can see that he has inculcated this thought that how he want to serve the society, the fellow Americans, or humanity as in whole. This is what researchers work towards all day and night. So you just mentioned, Dhananjay, that it was the self-reliance which brought you in research, and then making your own path is your mantra till now. How was the journey in PhD, and how that was different from your postdoctoral training? As you said, it's not a training, it's a work, so you have to deliver it. So how different is the research in two countries, like from India, and now you are in established researcher in USA?
Yeah, that's a good question. So PhD in India, you know, I can say it is like once you enter into the PhD in India, most of fellows, most of the student, they think about, you know, getting paper immediately and try to finish their PhD as soon as possible, like 3 years, 4 years, yeah, which is not a bad idea. Which is not, I'm not telling it's bad to complete your PhD within 3 to 4 years. That's well and good. If you are, prove yourself that you are well fit for the next step, like postdoc step in that 3 to 4 years, you are well and good. That I have no issues with that. But the thing is, what I faced difficulty is that even I joined to a renownable scientist. I'm talking about my boss. You know, so you even in my PhD, I didn't expect much from my boss. They just tell you just to do it, so you have to do it. Whatever you will, you get to you get a chance to learn from your boss in the initial period of your PhD, first 6 months or so. You have to perform. You have to perform from there. So
Mm-hmm.
like some techniques, you have to read yourself. You have to understand. You have to do yourself. Try to do things from your side. What
Mm-hmm.
what boss will tell, that boss will tell this thing. Yeah, you do this, you do that. So don't wait till the boss is saying. So you do something. Bring something, bring idea from your side. You know,
Mm-hmm.
and try to show your boss that see how is this? Does it work? Does it fit well? Then they will give the master plan. They will add the master plan into that. They will give the addition into that. So I think that the PhD student, should understand what their project is going to be,
Mm-hmm.
and try to read the basic. This is my, what I understood. Try to read the basic. Try to read a review rather than reading the multiple scientific paper. If you
Mm-hmm.
try to read review, you will understand means what? You will understand the thing very closely,
Mm-hmm.
because in a review everything would be there in that. So
Mm-hmm.
try to read a review paper related to your subject, and bring idea. It's not like your idea is bad, you're good. Bring some idea, bring some nobility into that.
Mm-hmm.
So then you can develop yourself. If you bring a thought into your mind, then you will definitely think how I will execute this. And then you will think about execute means plan. How will you plan this? And what plan means what experiment I'm going to perform here. So
Mm-hmm.
once you will perform this experiment, then once you've done the experiment, you being a self-critic. Don't feel that this is very common. Like when you used to get a Western blot data or something that we used to very curious. Yeah, we got the things. We are wow. Nothing. That's be critic. Be critic yourself 5 times. Why is
Mm-hmm.
this come? Why it is not that way? So critic yourself first. Don't be driven away by the result first time. So
Mm-hmm.
being a critic, so that critic will give you the sense of analyzing the data.
Mm-hmm.
That is more important, how you analyze the data. And once you analyze the data, then think about how I will put it into a frame or into a paper. So these
Mm-hmm.
all things needed. I think these all things we all the students, all the PhD fellow, they eventually they will learn in this during their PhD time course. So for
Mm-hmm.
me, this I passed through the same situation where I used to do myself things. Used to develop the technique myself. You can believe I have in my PhD I have mentored 16 to 18 students, the MS internship students.
Mm-hmm.
I have named 18 thesis the PhD intern student, 18 in my PhD. So my boss used to tell me that, yeah, you guide this student. So we used to do different types of cloning, different protein expression, different protein interaction. We used to do everything different way. So that's what came from your training. How you. Another other thing is that if you teach people, if you so the juniors have to do the experiment, eventually you will learn also. It's not
Yes.
like you will teach, you will also learn from them. And mistakes happen, so doesn't take mistake as uh, like the other way. Mistake happen and just learn from it. The other another third things I want to tell that I had no issue within my lab in terms of financial. I
Mm-hmm.
think we are the one of the richest lab in India that time. you
Mm-hmm.
know we have never faced any kind of lagging in buying the reagents, antibody, whatever you want. My boss just make it right. Okay, go with that. I think because they were that time they were the Wellcome Trust fellow, so they had a huge money in that. We had that time in I'm telling that time we had like 365 364 well plate, like the real-time PCR system, 384 well plate real-time system. So which I don't think any organization had such kind of facility. Everything, even I'm telling my friend that what I have seen in my PhD lab, I haven't seen in USA,
Mm-hmm.
because they came from USA. They knew, my PhD boss knew the what are the requirement to, you know, bring the science into that level. So you have to be self-sufficient with all these techniques, all these instrument. We have facts, we had a lot of things, everything we had. We never been to any other lab to do something. I don't know. I mean,
Mm-hmm.
we have never been to other lab to do a small experiment. No, never. So
Mm-hmm.
we are fortunate to have in that lab. And I saw some of your, like in your celebrity people, some I heard somewhere some people say that India doesn't have this facility. If you wait for long, you know, you have to wait something for long. Definitely you have to wait for a 4 to 5 months window, but it's not the same. The some labs are in India, they are more advanced than US. I can definitely say they're well
Mm-hmm.
equipped, well minded people. They're generous people, I can say. There are
Mm-hmm.
a lot of good opportunity, and the good brain, good scientists, as you can say. There's a lot of people that are there in India, and sometimes I can definitely compare the scientists of India as well as the USA scientists. At the same time, the lab also. So we
Absolutely.
had yeah, so we had never any difficulties doing science in in my PhD lab. So I'm very fortunate. I always thanks to my PhD boss that you have given such a wonderful opportunity to us. That's why we are here, and her blessing is there, I think. So yeah, that's what.
So now I would like to tell all the audience and viewers that, as Dr. Dhananjay mentioned, it is not about the if you are given a topic to do your PhD, you should have your own ideas. And that's what make it's your path, because you have to create your own path and walk onto it. That is for PhD. But considering about the limitations of the finances of the laboratory is also one of the very important point when you think that you want to pursue a PhD. Just have a look on how well funded the lab is, because as Dhananjay is sharing his journey, he never faced any limitations, which in some of the labs are still in there. So you have to understand your idea, your thought, that the lab you are going to join for your PhD, how well funded they are, how what are the potentials, because they might get a grant in very recent very recent future. So before you join, and you just don't join a lab because they don't have funding, but if you are in conversation with the PIs, with the investigators, the scientists, the professors, and they tell you that okay, the grant is about to come, so you have to decide, make all these calls yours, because it is, you want something, and always a mentor want a great mentee. As Dhananjay mentioned multiple times that he's fortunate, he was lucky to have them as their mentor. They will also share the same feeling, that they were fortunate enough to have Dhananjay as a PhD student in there, and that's how it goes both ways. So Dhananjay, when you moved to United States for pursuing better research, and now you have established the early career. Now, when we see what are your future plans?
Yeah, as I said, I have no plan.
Mm-hmm.
You know, I believe in hard work, and I
Mm-hmm.
do that. I'm saying that, I mean that. You know,
Mm-hmm.
I'm I want to tell your viewers that it's depend upon how how you work, how much effort you are putting on your work. You know, it's not that, is there some people are there, if uh, like they work little but they get more things, like in terms of paper, in terms of grant. uh, yeah. So mine is that, like, I believe in hard work. Always I do that hard work. Even Sunday, I never in since my 5 6 year career at USA, I don't think I have ever taken a leave in from Monday to Sunday. I'm always there in the lab. You can see still I'm on the lab, you know. So lab doesn't mean that you can come and watch movie in the lab. Some people used to do that, but it's not that the case. So the case that if you're putting your effort 100%, and that 100% definitely give you a 30 to 40% output. I'm not telling your 100% effort will give you 100% effort result, but definitely to give you 30 to 40% results. But if you can continue this with 30 to 40% results like every week, so you see how your effort will be in a 1 year span or 2 year span.
Mm-hmm.
The other thing is that hard work, the same time as I told you to put your thoughts in what you are doing, and what you are trying to see, what you're trying, what the question in front of you, and how you are trying to solve that question. That is the most important. And at the same time, you have to find something very interesting. How it be interesting? I would just give an example. Some people try to escape the easiest route.
Mm-hmm.
I'm not that way. I don't want to go through the easiest route. Let it come, let go into the deep of that. It's like a rabbit hole type. Just
Mm-hmm.
find something new, go into that, go deeper and deeper and deeper, try to find yourself, where science is very vast. If I
Yeah.
talk about the biology, you cannot be a master of anything, everything, sorry. You cannot. I'm doing cancer biology, I cannot be a master of immunology, I cannot be master of bioinformatics, I cannot master of metabolism.
Mm-hmm.
Now I'm doing a signaling biology, epigenetics. Definitely I have to be master of that. So my boss always tell that, what my my postdoc boss Durden, he used to tell me that you do one thing and involve in the one thing.
Mm-hmm.
Don't involve a lot of things. If you
Mm-hmm.
want involved in one thing, you will get recognition, you will get respect. If you
Mm-hmm.
think that I will do this, I will do that, some people used to do to get papers, to get something, but you won't be get respectable,
Mm-hmm.
because you have half knowledge on that.
Mm-hmm.
So that's what I try to go most deeper and deeper. I don't want to escape the easiest through the easiest route. So that's my way of doing science here at USA. Although the paper, the grant, that is a different sort of thing. When the paper will come, that will come. But you should like to enjoy the path. Just
Yes.
find something, go deeper, and you will enjoy. Like
Nice.
scuba diving, you can say.
Absolutely. I believe that science, the only way you can be a researcher for your lifetime is if you can enjoy it, if you love it, that
Yep.
without taking leaves for 5 years. Dr. Dhananjay has done exceptional work. I would like to tell all of our viewers that it is your call. If you enjoy it, we had researcher celebrities who don't believe in because so work-life balance is something which people talk about, and our researcher celebrity says that they don't see this as work-life balance because research is their life. So they say it is just life all the time. Be inside the lab or be outside the lab, they just enjoy it uh, the same way, and that's what makes you this passionate researcher that you can go to the lab at any time or at any day also.
Yep.
Now we walked at the edge of our conversation timing. I would like to tell all of our viewers that now you have heard Dr. Dhananjay's view on how a researcher life is. And if you want to ask him anything, you can write to Dr. Dhananjay directly. Or if you want Empowering Science Foundation to put you in contact with our researcher celebrities, please do write to us and then we will put you in touch with them. With this, we will say thank you, Dr. Dhananjay, for taking time out
Thank
of
you.
your busy schedule. As you said, you are still in lab and doing work. So it is great to have you as our researcher celebrity, and we wish you all the good luck for your future.
Thank you, Dr. Kuswa, for having me here. I think one thing we have left, I think
Mhm.
uh, the scientific part, I think we didn't discuss.
Yeah,
Uh,
because you just explained every single part in this conversation, that your suggestions and what you suggest to all of our viewers, how should they follow. because when you said making your own path, following your own, generating your own idea, and doing what not everyone does, this is what makes them a true researcher in all the core. So thank you very much, Dr. Dhananjay.
Yeah, just and I will just tell to your viewer in 2 sentences. So,
Yes,
so
please.
I didn't elaborate my postdoc work or what I am doing right now. I just elaborated in to 1 to 2 minutes that I'm working on signal biology and epigenetic gene regulation
Mm-hmm.
and hematological malignancies like the B cell related cancer, that mantle cell lymphoma and chronic lymphocyte lymphoma. And recently, I'm working on the neuroblastoma thing.
Nice.
And the thing is that the only one thing I would just
Mm-hmm.
just tell to your viewers that
Please.
so why my work is little helpful for the society.
Mm-hmm.
So just I'll give you an example. So if something happen at your home, suppose short circuit happens at your home, so what you
Mm-hmm.
will do? Will you switch off your nearby switch where you are sitting, or you will switch off the main switch? What you will do? So
Mm-hmm.
to get rid of the short circuit, I think it would be a better idea to shut up from the main switch, you know,
Mm-hmm.
and you will see that you will get rid of all the short circuit things. Then you will take a look at where the short circuit happens, and then you will rectify it. The same
Mm-hmm.
similar way, the cancer cell regulates. Uh, the cancer cells survive due to the lot of wiring system. That's called the signaling. So
Mm-hmm.
if something happens, why the drug getting resistance to these cancer cells? So you have
Mm-hmm.
to see, you have to stop the main switch. So in our term, the main switch is called the central node. Thus,
Mm-hmm.
if you control the central node in our term is the PI3K-AKT signaling. That's what we are working on. If you control
Mm-hmm.
the PI3K-AKT signaling, then you can get rid of most of the signaling, the downstream signaling. And
Yeah.
at the same time, if you regulate the epigenome, like the
Mm-hmm.
enhancer, promoter, or super enhancer activities, definitely you will get rid of somehow you will get rid of the cancer disease. And that is our main focus, to design uh, to understand the resistance mechanism and to develop some small molecule inhibitor to capture this signaling pathway as well as the epigenetic pathway. So then I think it can be a remedy for the cancer patients.
That's brilliant. And thank you very much, Dr.
Mhm.
Dhananjay, for sharing this. And now all of our viewers are free to write it to you and to us.
Yep.
And with this, good luck for your future research. Thank you
Thank
very
you.
much.
Thank you very much for having me.